Welcome to our exploration of the pivotal experiments that led to the groundbreaking discovery of the structure and function of DNA, the molecule of heredity. In the early 20th century, the precise nature of the genetic material was still a mystery. Scientists were divided between proteins and nucleic acids as the carriers of genetic information. It was not until a series of critical experiments that the picture became clearer. The first important milestone was in 1928, when Frederick Griffith discovered what he called the transforming principle during his work on Streptococcus pneumonia.
In the world of molecular biology, I, Frederick Griffith, have left a notable imprint. Back in 1928, while working as a bacteriologist, I became intrigued by Streptococcus pneumonia, a bacterium causing pneumonia. There were two strains of this bacterium I focused on. One had a rough appearance under the microscope, which I called the rough or R strain, which was non-virulent. The other, known as the smooth or S strain, was virulent due to a protective capsule. In my experiments, mice infected with the S strain developed pneumonia and died, while those with the R strain survived. The protective capsule of the S strain was the key factor here. Then I decided to heat-kill the S-strain bacteria. This process destroyed their virulence, or ability to cause disease. I then mixed these heat-killed S-strain bacteria with living R-strain bacteria and injected the mixture into mice, expecting them to survive. To my astonishment, the mice died. Upon examination, I found living S-strain bacteria in their bodies. But how could this be? It appeared that the R-strain bacteria had acquired traits from the heat-killed S-strain bacteria, transforming into the S-strain. I termed this the transforming principle. Although I could not identify the nature of this transforming principle, I hypothesized it was a certain component from the heat-killed S-strain that had caused the change. My work paved the way for Avery, MacLeod, and McCarty, who later confirmed that this component was DNA, making a huge leap forward in our understanding of heredity.
The real breakthrough came in 1944 with the experiments of Oswald Avery, Colin MacLeod, and Maclyn McCarty. Building on Griffith's work, they were able to demonstrate that DNA, not protein, was the transforming principle, indicating that DNA is the genetic material.
Hello everyone, we are Oswald Avery, Colin MacLeod and Maclyn McCarty. Today we'd like to guide you through the journey of our work, where we unveiled DNA as the molecule responsible for carrying genetic information. Our quest began from the mysterious transforming principle that Frederick Griffith discovered. But what was this transforming principle? That's the question we set out to answer.
Our strategy was simple but effective, a process of elimination. We extracted different components from the heat-killed virulent bacteria proteins, RNA and DNA, and added them individually to the harmless bacteria. Our goal was to observe whether transformation would occur. Proteins were the leading candidate at that time, given their variety and complexity. However, much to our surprise and contrary to prevailing beliefs, adding proteins to the harmless bacteria did not trigger any transformation. Neither did RNA. But when we introduced DNA to the harmless bacteria, we noticed that they had turned virulent. This suggested that DNA was the elusive transforming principle.
To further corroborate our findings, we conducted another set of experiments. We used enzymes to degrade the different components of the heat-killed bacteria. When proteins and RNA were broken down, the transformation still occurred. However, when DNA was broken down, the transformation failed to happen. This was the decisive moment, confirming that DNA, not protein or RNA, carried the genetic information. Our work was initially met with skepticism, as it was hard for many to accept that DNA, with its seemingly simple structure, could carry the complex information that defines life. However, our results were robust and reproducible. In the end, it was the strength of our evidence that convinced the scientific community.
In the 1950s, Work by Rosalind Franklin and Maurice Wilkins using X-ray diffraction techniques provided the first clues to DNA's double helical structure. A photo known as Photograph 51, taken by Franklin, provided critical information about DNA's structure. Finally, in 1953, James Watson and Francis Crick proposed the now universally accepted double helix model for DNA, built on the principles of molecular biology and the critical insights from Franklin's Photograph 51 and Chargaff's work detailing the pairing of bases. They suggested that the DNA molecule consists of two strands of nucleotides arranged in a specific manner.
Greetings, I am Rosalind Franklin. During the 1950s at King's College London, I was using X-ray crystallography, which shows the molecular structure of crystallized materials. Using this, my student Raymond Gosling and I captured an image, now famous as Photograph 51. This photograph, showcasing an X pattern, was a crucial hint towards DNA's helical structure. Interestingly, DNA has two forms, A and B. While initial work with the A form was unclear, the B form yielded Photograph 51, suggesting a double helix structure for DNA. Unknown to me, James Watson and Francis Crick had access to this image. With its insights, they proposed the double helix model of DNA. They elucidated that the DNA molecule consists of two strands coiling around each other, where bases like A pairs with T and G with C. Regrettably, my contributions went largely unrecognized during my lifetime, overshadowed by Watson and Crick's model. Today, however, my role is acknowledged, highlighting the essence of collaboration and recognition in scientific endeavors.
Thus, these pioneering experiments paved the way to understanding DNA's structure and function, revolutionizing the field of genetics and leading to a myriad of advances in our understanding of life and its processes.